My name is Momchilo Gavrilov, and I'm Sharp Diagnostics co-founder and chief scientific officer. Our company, Sharp Diagnostics, started in Baltimore, very close to Johns Hopkins University, where I was a postdoctoral fellow, engineering various proteins for biotechnology applications. Our company, Sharp Diagnostics, develops the next generation viral load test based on engineered enzymes. And our mission is to bring fast and quantitative diagnostics directly to the patient and the provider. And here, I'm taking this opportunity to look for a business mentor and help to raise 1.5 million to build a prototype of a quantitative HIV-1 viral load test and enter FDA trials in 2025. So first, let me introduce you to this problem. As you probably know, about 1.3 million people live with HIV in the United States. And pre-pandemic, about 2.5 million HIV tests were conducted each year. Now, these tests can be divided into two major categories. There are qualitative HIV tests. They're typically interpreted as either positive or negative, and they're meant for people with an unknown HIV status. These qualitative tests are available at the point of care or even home. And then on the other hand, there are quantitative tests. Quantitative tests, they provide a viral load value in terms in copies of HIV particles per ml of blood. These tests are specifically used for HIV positive people. They're used to monitor the disease progression, measure the treatment efficiency. These tests also help people maintain their undetectable status and can help protect others from HIV. Quantitative tests are typically repeated every 3 to 6 months. They require multiple visits to the healthcare provider for a consultation, then a lab for a blood draw, and again to a healthcare provider to adjust the therapy. And they typically are associated with the long sample to result time that can take even several days. Now, our solution actually eliminates these three problems. It minimizes the need for visits to a healthcare provider, and it also provides sample to result in less than 30 minutes. So our solution is Sharp Diagnostics quantitative point of care HIV-1 test. Our test starts with a finger prick, where typically 20 microliter of blood is collected. Then the blood is incubated in the analyzer device for about 30 minutes. And at the end of that time, the device provides the value in copies, number of viral copies per ml of blood. So the pipeline for obtaining the value is pretty similar to, for example, a glucose monitor. And having this technology actually enables viral load monitoring at the point of care, and also enables future HIV-1 quantitative home tests. And that, of course, reduces the number of visits to the healthcare provider. This tool also opens many possibilities for telemedicine options. Since we draw a small amount of blood, our test is suitable for infants, and the test generally has a very small footprint, and the consumables can be easily disposed. Now, the reason why we can actually do this test nowadays and not earlier is because since the pandemic started, there were huge investments in molecular diagnostics, and we have developed fundamentally new technology that minimizes the footprint and enables this type of a test. So the existing quantitative tests used a PCR technology developed in the 1980s. And this technology, PCR, requires these big and bulky thermocycler machines, and PCR must be carried out in a sophisticated laboratory to accommodate this equipment. So what we have done at Johns Hopkins, we actually engineered a molecular motor, an enzyme, to carry out PCR in a single tube rather than having this instrumentation. So the idea behind our technology is to use an engineered enzyme to split the DNA at constant temperature in a tube, rather than using this bulky machine to periodically heat DNA. And that technology actually enables us to shrink down the size of a test to a cell phone and enable these quantitative molecular diagnostics at home or the point of care. And maybe it's worth mentioning that, for example, for COVID, we can only do antigen tests at home, but for COVID PCR tests, one actually has to go to a clinic and give a sample there, so the test can be carried out in the lab. So our technology, the basic technology, it has been validated in a peer review process. And about 8 months ago, we published a manuscript describing how this engineered helicase enzyme can actually replace the thermocycler while it maintains all the desired characteristics. And our patent, where I'm one of the co-inventors, have been submitted by Johns Hopkins Technology Venture Office. In the next slide, I'm comparing our basic technology with other competing technologies, PCR, antigen, LAMP, SHARP, and HDA. And here, essentially only SHARP allows both quantitative tests and portable point of care tests, while other technology might be even too expensive to be carried out at point of care. Now, what is our product? What is our vision about the product? Our vision is to be offering a test analyzer and a consumable with a razor blade model. The analyzer device will display the value and analyze the signal coming from the consumables, and the consumables will contain a proprietary enzyme mix to carry this reaction. Since this is an enzyme-based technology, it has a very low manufacturing cost. So there are similar technology that use microfluidic chips and these engineered devices, and they tend to be very expensive. While since our technology involves only enzymes, the cost to manufacture one of these tubes is very low, and we can even offer this technology at 10 times lower price than the existing lab-based quantitative tests. Now our plan is to develop, to start the FDA trials in following phases. First, we will have to establish the enzyme manufacturing because that is essential so we can start doing tests with the manufactured enzyme. Then in the second phase, we will prove that our enzyme can actually identify and quantify number of HIV particles in a blood. In the third phase, after we confirm tests from the phase 2, we will develop and integrate this hardware analyzer device. And then we expect in the mid-2025 to actually approach FDA and slowly enter their trials. Now, here's our team. I am one of the co-inventors of this technology and a former postdoctoral fellow at Johns Hopkins. And then Taylor, he's our CEO. Taylor just graduated from Johns Hopkins in a program in biochemistry and biophysics. And then our scientific advisor is Professor Tejib Ha, who is also my postdoctoral advisor at Hopkins. And we are currently actively looking for a business mentor and advisor to support us in this project. And finally, I just want to give you again our mission statement. Our mission is to bring quantitative viral load tests right to patients. And if you have any further questions, please ask, or you can contact me using my email or phone given below. Yeah, thank you.